Hello dear friends, welcome to study.official. This is our next video and we have chosen the topic of crop physiology in this. Let's deal with some of the topics of crop physiology, that is the plant growth regulators. In this class we shall continue with this topic. As first of all, let's see the contents. As contents, what are it? The introduction to plant growth regulators, introduction to auxin, biosynthesis, occurrence of the auxin, mode of action of the auxin, physiological roles of the auxin. These are the contents. Let's continue. Okay, let's see what our plant growth regulators are. One by one, let's see the definition of it. That is hormones. Plant growth regulators are the hormones, first of all. In general sense, plant hormones means, here hormones means plant hormones. It's an endogenous compound, means it is within the plant, which is synthesized at one site and transported to another site where it exerts a physiological effect in a very low concentration. That is, these are very effectful in a very low concentration and very harmful in higher concentration. So these functions are seen only in the low concentration, that is, they are present in, they are synthesized in one site and they will be transported from that synthesized site to exert its physiological effect. But exceptionally, ethylene is a plant hormone which is not transported, which shows effects where it is synthesized. Next, see plant growth regulators. Plant growth regulators, these are the organic compounds other than nutrients, that is some other than nutrient compounds that affects the physiological process of growth and development in plants. These plant growth regulators affect the growth, they increase or the fruiting, and many functions are there. They will be affected in plants when applied in a low concentration. Yes, if we compare the definition of plant hormones and definition of plant growth regulator, they are a bit same. But dear friends, please remember that the main difference between the plant hormones and plant growth regulators is plant hormones are purely synthesized within the plant. Plant growth regulators are includes the plant hormone and synthetic one, or artificial one and natural one. Okay, let's continue. Oh, there's a scientist who has given some of his opinions, I say ideas about the plant growth regulators. His name is Julius von Sachs. He proposed some mechanism, that is, a chemical messengers are responsible for the formation and growth of different plant organs. To develop some of the plant organs, it is compulsorily there are some chemical messengers are required. Such chemical messengers play a major role in the activity, the interactivity. There are many mechanisms that happens in the plant, that for that these chemical messengers are helpful. And he also said that gravitation could also affect the distribution of these substances. Sometimes these substances distribute within the plants and the gravitation affects such a distribution. Next friends, auxin. In this class we just learn mainly on auxin. What are auxins? That is the substance promoted the elongation of the coleoptile section. It was eventually named as auxin from Greek word auxein, meaning to increase or to grow. See friends, these are first discovered on the coleoptile part, that is by the Charles Darwin and his son Francis Darwin. There are many systems and how the discovery took place. In this class just we shall know it has been discovered and it is the main reason for the growth and development of the plant. So from they took a Greek word, means to increase or to grow, that is auxein word, and they named this auxin. And the term auxin was introduced by Kogel and Hagen Smith in 1931. This term was introduced at that time. Okay, next important is the biosynthesis of auxin. How the synthesis of auxin takes place in a plant in a natural way. There are three proposed mechanisms. One is the indole pyruvic acid pathway, tryptophan pathway, indole acetaldehyde pathway. One by one we shall see. In this I have tried to explain both mechanism, that is indole pyruvic acid pathway and tryptophan pathway, in a single image. The basic or the important precursor for the development of the auxin is tryptophan. It is compulsory and it is the precursor. Other than this, the auxin can't be synthesized. Okay, from this tryptophan, first we shall see indole pyruvic acid pathway. In that, first what happens? Tryptophan converts into indole 3 pyruvic acid from the enzyme. The enzyme that acts is tryptophan transaminase. Here the release of NH3 takes place, that is deamination takes place. See here, deamination takes place, and due to this deamination, the indole 3 pyruvic acid formation takes place. And from this indole 3 pyruvic acid, it leads to indole 3 acetaldehyde. Here decarboxylation takes place. They have amine and carboxylation. So decarboxylation takes place, that is release of CO2 takes place in the presence of the enzyme indole pyruvate decarboxylase, and leads to the formation of indole 3 acetaldehyde. And this indole 3 acetaldehyde is common for both indole pyruvic acid pathway and tryptophan pathway. From this, the development of the indole 3 acetic acid takes place in the presence of indole acetaldehyde dehydrogenase enzyme. That is indole pyruvic acid pathway. Tryptophan, in the tryptamine pathway, tryptophan converts into tryptamine. Here the release of carbon dioxide takes place. In the indole pyruvic acid pathway, first NH3, that is deamination, takes place. Here first decarboxylation takes place. Just a simple difference between these two pathways. If we remove NH3, then it's indole pyruvic acid pathway in the beginning. If we first remove the CO2, then it is tryptamine pathway. This takes place in the presence of tryptophan decarboxylase. And tryptophan decarboxylase leads to formation of tryptamine. And this tryptamine will convert into indole 3 acetaldehyde by the release of amine group, that is NH3. Deamination takes place in the presence of amino oxidase. This indole 3 acetaldehyde leads to formation of indole 3 acetic acid, that is the natural auxin IAA in the presence of indole acetaldehyde dehydrogenase. These are things you should remember, you should by heart the things, but in general, tryptophan converts into indole 3 acetic acid by the release of amine and carboxyl groups. And there is another important pathway, that is indole acetaldoxime pathway. In this, friends, in the beginning I said the precursor is tryptophan, and in the tryptophan it converts into indole acetaldoxime, then to glucobrasicin, then to indole acetonitrile, then to indole acetic acid. This is the complete pathway, that is the formation of natural auxin, that is indole acetic acid IAA. Okay, we have completed the biosynthesis. Let's see the occurrence. Where all this auxin occurs? It occurs normally on all parts of the plant body. All parts of the plant body produce auxin, and the major sites of synthesis are shoot tip, developing seeds, and buds. Shoot tips are the where the actual growth takes place. Amount of auxin is very high in the tip part, that is where importantly the growth takes place. And from the tip, the concentration of the auxin start to decline. And the example for the natural auxins are IAA, indole acetic acid, and 4-CL, IAA, PAA, IBA, indole butyric acid, indole propionic acid. These are all natural auxins that are available. Next, availability of natural auxins are in two ways. The availability in the natural way, that is within the plants, the availability of auxin is in two ways, that is on the free auxins or bounded auxins. In the free auxins, dear friends, which freely move or diffuse out of plant tissues readily, that is it can move, it will be synthesized and move towards various parts where the requirement is there, these free auxins move. But bound auxins will be, they are not free, they are not active compounds, they are yet to convert into active compounds. That is, after some enzymatic action, that is hydrolysis, autolysis, or enzymolysis, These bound auxins, these auxins which will be in inactive state will be converted into auxin stage. That's active auxin stage. Free auxins are initially released in a active auxin stage. Okay, after we have completed the natural auxins, now the synthetic auxins, and they are produced in various different mechanisms, but just we have tried to list some of the synthetic auxins, that is IBA indole butyric acid, NNA naphthalene acetic acid, methyl ester of naphthalene acetic acid, TBA 2,3,5 triiodobenzoic acid, MCPA 2 methyl 4 chlorophenoxyacetic acid, 2,4-D 2,4 dichlorophenoxyacetic acid, 2,4,5-T 2,4,5 trichlorophenoxyacetic acid. These are some of the synthetic auxins. Okay friends, let's see what is the mode of action. Mode of action of this auxin. First, the important mode of action is the acid growth hypothesis, which is proposed by Hager, Menzel, Kranz. These propose this mechanism. Mode of action means, friends, how the auxin, that is in general sense we say they increase in inside the sort the cell wall and all. What might be the possible hypothesis of this action? That is first acid growth hypothesis, which was proposed by these three scientists. And here let's see what happens, friends. Hydrogen ion decrease pH value and break acid labile bonds or active wall hydrolyzing enzymes. See, you can see here in this auxin stimulate the pumping of these H plus ions. These H plus ions in the cell wall, what they does is they decrease the pH value. This is if there are more H plus means acidic condition, right? So acidic condition increases, pH value decreases, and this acidic condition breaks the acid labile bonds, or we can say activate wall hydrolyzing enzyme. Wall hydrolyzing means the enzyme that break the cell wall or activated or by this hydrogen ions, that is the acid condition. And after breaking the strong cell wall, then automatically they undergo softening. The cell wall, the hot cell wall becomes softened, and after once the cell wall is weak, then automatically the expansion, the elongation of the cell takes place. This is the major hypothesis. In this image, see, this is they have shown here. Auxin stimulates to pumping this H plus ions to cell wall, where it converts into acid condition that leads to the break of cell wall and leads to the cell expansion. There are some other possible hypotheses for the cell expansion, that is cell elongation may be due to increasing osmotic pressure and permeability of cytoplasm to water, and decreasing the cell wall pressure. That is, the increasing the osmotic pressure and permeability of cytoplasm to water. Once it is under pressure, they automatically get weakens and the water might enter by decreasing the cell wall pressure. That is another hypothesis for the formation or the cell expansion. There is another important hypothesis that might be possible here. Auxins stimulate the production of hydrolyzing enzymes like B1, 3-gluconase, pectin methylesterase, cellulase, which soften the cell wall and increase the plasticity, resulting in reduction of wall pressure and cell elongation. There, they try to pump the hydrogen ion and further process takes place, but here it directly auxins stimulate the production of wall hydrolyzing enzymes, that is B1, 3-gluconase, pectin methylesterase and cellulase and all. These are the important mode of action, that is how the cell expansion might takes place. Now after the cell expansion, some enzymatic activity or mechanism should take place to expand, right? That is, under the influence of auxin, cellulose synthetase increases and new wall material is synthesized. After once the cell wall and they are weak, the new wall materials are synthesized from the cellular synthetase, and within the cell thus resulting in the extension of or growth of the cell. Next, what are the physiological roles? There are many roles these auxin plays in many plants. Cell division, initiating and promoting cell division in certain tissues like cambium. See, in this image, I have tried to show you, means the green color here is a cambium tissue. This is a cambium tissue, friends. This is normally there in the trees. Once there is a secondary growth, once it can gain the cell dividing capacity, and such a dividing capacity is induced by this auxin, this cambium, which leads to secondary growth, that is lateral growth, means year after year you can say how finding the aging and all this growth, some growth capacity are regained due to the action of this auxins for this cambium. Next, wound leads to callus. Yes, if there is a wound in a plant, it leads to callus formation due to the proliferation of the parenchyma cells, which can stimulated by auxinate chemical traumatic acid. See, in the parenchyma, that is the callus formation. See here, the callus formation takes place due to the proliferation of the parenchyma cells. That is due to the either due to the proliferation of the parenchyma cells, which can be stimulated. This callus formation is stimulated. This callus formation is stimulated either by auxin, or we can artificially induced callus by treating with a traumatic acid. And dear friends, after the formation of the callus, they convert into very productive part, and thus the callus formation is very important to join two plant parts. Like it is mainly useful in the grafting, friends. When we join the rootstock and scion, there should be the connection between them, right? So to form a connection between them, first the callus should form. To form the callus, the auxin plays a major role. Yes, we economically use these in the grapes, grafting up the grape, grafting immersion of stalk and scion in 0.1% of IAA. Before grafting, we just immerse them in the IAA in the 0.1 concentration. Next, root initiation, friends. Root initiation. Stem cuttings of some plants readily form adventitious roots when put in the soil. Normally, if there is a stem cut, if you just plant them in the soil, if every condition is good, they just undergo rooting. But in some plants the stem cutting does not lead to rooting. That is, here rooting means first the adventitious roots should form, which are very important to hold them strongly, firmly. Then the main roots will start forming. These adventitious roots are not common in every stem cuttings. In some we have to induce, that is the induction of this adventitious roots is by this auxin. We use commonly the NAA or IBA, 500 ppm of IBA in air layering. Air layering, you have to initiate the root, right? So there you can use. Herbaceous are more responsive than woody perennials because herbaceous actively divide, right? So they are more responsive than woody perennials. Okay, we have seen very important two roles. Now we shall see the another very important apical dominance. See friends, this auxin is mainly present in tip. I have said in the beginning. This is a problematic in some condition. That is, what happens? They mainly accumulate. Just see the image. They mainly accumulate in the apical bud. If this apical bud is more active, when the auxin is present, more cell division takes place. Then automatically this will grow. If this is the very active, then the lateral parts won't be active much. Lateral parts will be weak. So the apical part is dominant over everything. We call them as apical dominance. Normally we don't favor this apical dominance. We just cut off the tip. In some plants we have to induce this apical dominance because the above land, that is the vertical growth, will not be favorable in is not present in some plants. There we have to use auxin to induce the apical dominance. Okay, the potato and tomato fails to suppress the lateral buds due to lack of auxin and form branched or bushy plants. In such condition, we have to apply the auxin. The phenomena of inhibition of the lateral buds by the shoot apex is known as the apical dominance. That is the definition of it. Next, another important function, that is the inhibition of abscission layer. Dear friends, abscission means the process of dissolution of the middle lamella and primary cell wall of the cell at the base of the petiole, pedicel or peduncle. See, this is a base petiole, pedicel or peduncle part. This is a base part. See here, the abscission has taken place here. That is, the middle lamella and primary walls here are getting weakened, and they led to the detachment of this complete part from this. They lose the connection. Such a condition is known as abscission. Balance between the inhibition of auxin and promotion of substances like ethylene and ABA. Ethylene and ABA, they just lead to the aging. After once they age, they will fall off. So the lack of the auxin means the active growth will not be there. If the active growth it won't be there, then it will automatically lead to aging. That is the balance between the inhibition of auxin and promotion of ethylene and ABA. Both take place simultaneously. There will be a balance between them, thus lead to the abscission. So it should not be, the both conditions should be in a balanced condition to prevent this abscission. Several auxins inhibit the abscission. Auxins, by inducing the growth activity by the auxin, that leads to the prevent the abscission. In the economical point, we can use NAA at 20-30 ppm twice at 15 days interval. This reduces the flower and fruit drop in chilies and cotton. 2,4-D prevents defoliation in cabbage, cauliflower that often occurs during the harvest. The next important function is flower initiation. Normally, friends, the auxin inhibits the flowers in many plants, many species, but exceptionally in a day neutral plant. That is, in a pineapple, in the day neutral plant, this auxin induces the flowering. They bloom due to the application of NAA or 2,4-D. The effect is due to auxin mediated through ethylene formation. This auxin leads to the formation of the ethylene, which leads to flowering. That is the main, maybe the main cause, but exceptionally this has occurred. Application of auxin alters sex ratio in flowers. In the monoecious cucurbids, difference in the monoecious cucurbids, there is increase in number of female flowers but decrease in number of male flowers on the application of the auxin. But in dioecious like cannabis, male plants produce female flowers, but effects are mediated through ethylene formation. Here also effect is through the ethylene formation. In the male plant, only male flowers should produce, right? But female flowers are produced by the application of the auxin. Okay, next, the production of the parthenocarpic fruits. In an unfertilized or in the absence of pollination, there is no pollination, and in the unfertilized condition, the application of the auxin causes the development of ovary into the fruit in several plants like tomato, brinjal and others. Means they don't undergo fertilization and all. The ovary that converts into and develops into a fruit. Such condition we just say, if there is no fertilization and develop into a fruit, we say parthenocarpic fruit, and the auxin induce such parthenocarpic condition. Next, eradication of weeds. In the beginning I have said that this should be in a micro quantity. If it is not in a micro quantity, there will be a problem. Maybe toxic to some plants. Yes, in some plants we have used it as a weedicide. Yes, 2,4-D. I think you have heard about the 2,4-D. This 2,4-D acts as a weedicide. They acts as a weedicide. See, in the very high concentration of the auxin, it overstimulates the growth. Means hyper growth activity will be there of root cells. In the root cells, hyper growth activity will be there, thus resulting in the destroyed or distorted roots, where the blockage of sieve tubes takes place, where the food should transport, and blockage of sieve tubes leads ultimately decay and kill the plant. and the 2,4-D and 2,4,5-T are effective weedicide at 1 to 3%. Dear friends, the auxin should be in a very minor quantity. If it is in a high quantity, then it leads to very damaging state. 2,4-D is selective. It's highly selective and highly toxic to broad-leaved plants and or dicots. For dicots, it's highly selective and toxic, and relatively non-toxic to narrow-leaved plants. In some narrow-leaved plants or monocot, they should be they are non-toxic, much non-toxic. So okay, next important is the growth in thickness. Secondary growth increase in thickness of stem and root due to radial growth. Means that is what I have said in the beginning. The cambium gain its importance, gain its capacity to redivide and thus lead to the increase in the thickness. Next, vascular differentiation. Vascular differentiation of the cambial derivatives into xylem and phloem under control hormones involve interaction of both auxin and gibberellic acid. Differentiation of the cambial. Once the cambium divides, the cell those get from the cambiums will differentiate with the action of this auxin and gibberellic acid. They differentiate into xylem and phloem that are maintained through this auxin. It's also one of the important part. And prevention of lodging. Friends, naphthyl acetamide, when applied on the base of oats and flax, lodging is prevented. See, some of the functions of these hormones, auxins, or any plant growth regulators, they will not be uniform in every plant species. They differ. Some might be useful in some plants, some may not be. See, the weedicide 2,4-D, it is very effective in dicots, but not much effective in monocots and all. Such type, here also another condition where lodging is problem in oats and flax. There, by applying the naphthyl acetamide, one of the auxin, they prevented the lodging condition. This one of the helpful in some cases. Let's see how the inactivation of this auxin in plant takes place. Auxin destructed by enzyme IAA oxidase. IAA oxidase, they just destruct this auxin. That is, see the mechanism, that is the reaction. IAA with the H2O2 peroxide plus oxygen, IAA oxidase acts the enzyme, then leads to the 3-methyl oxindole with the water and carbon dioxide. Thus the inactivation of auxin takes place. Thank you friends, thanks for listening to this class. I hope it might be very helpful. I think it might be a little lengthy, but these are very important for you to understand and learn the things. Please like, share, comment and subscribe to our channel agristudy.official. Follow us on Instagram agristudy.official, Facebook agri/study, YouTube agriculturestudy.official. Please subscribe. Thank you. Let's meet again.